Before we start the formal presentation, I have a few items I would like to share with everyone. Our Crops Extension Specialist team put on winter webinars each year like this one. We work out of 10 different regional offices across the province, and each office contains a wide variety of specialists that can help you with any agricultural-based questions. To connect with our specialists, please call your local regional office or the Sask Ag or the Ag Knowledge Center provided on the screen here. There are 8 agri-arm sites located throughout the province that are dedicated to bringing the industry Saskatchewan-specific information and demonstrations on new and existing agricultural practices. If you're interested, please follow the QR code on the screen. This is my personal favorite. If you have ever heard the words on the radio, Saskatchewan is 98% complete. Our harvest is 98% complete in Saskatchewan. You've likely heard of the Saskatchewan Crop Report. Myself, along with my colleagues here at Sask Ag, work to write the crop report each week during the growing season to bring the industry updates on what's going on in the field. So we rely on a vast network of crop reporters, which includes producers, agrologists, and other members of the ag industry. So if you have any kind of an agricultural background, I highly suggest that you sign up to be a crop reporter. The surveys only take about 5 to 10 minutes each week, so please consider signing up because your information does a great service to the Sask Ag industry. I also want to mention one of our flagship events we put on, which will be the 2026 Crop Diagnostic School, which will be hosted in Swift Current, Saskatchewan this year on July 29th and 30th. More details on the event are coming soon, but along with Crop Diagnostic School, the local agri-arm sites also have field days coming up, so stay tuned for those. Please note that our upcoming schedule of crops winter webinars, but some of our other Sask Ag teams have a webinar series of their own. So the weed management webinar series is put on by our range management specialists and agri-environmental specialists. So register today on our website at the Sask Ag events calendar. Lastly, I just want to make sure that everyone is aware of the funding available through the Sustainable Canadian Agricultural Partnership. You can reach out to your local agricultural program specialist or the Ag Knowledge Center to find out what funding you may qualify for. Along with this information, we have additional resources for everyone under your handout section in the chat box area of the toolbar. These will be shared after the webinar along with the recording. After the presentation today, we will have a live Q&A session with the presenter. The main hub for today will be your webinar screen, and as mentioned, we will share any relevant downloadable handouts with you in the chat box area. The entire session will be recorded, so if you happen to miss anything or want to revisit any part of the presentation, you will be able to do so. Recording links will be sent out either later this week or early next week in a follow-up email. An evaluation will pop up at the end of the webinar this morning. Please ensure that you fill this out, as these will help us determine what topics you want to learn more about and help us prioritize research needs for the future. We have a few housekeeping notes to keep in mind during the webinar today. If anybody has questions during the presentation, you might type them in the question box on the top right-hand corner of the control panel. We will get to as many questions as possible, and your questions will remain anonymous and will only be sent to me. During the presentation, you will be muted so that we can ensure that there's no feedback that will interfere with the audio. So now let's get started with today's presentation. We have Dr. Kelly Turkington joining us, who is a research specialist and plant pathologist with Agriculture and Agri-Food Canada in Lacombe, Alberta. His research focuses on cereal and canola disease as a part of collaborative studies with colleagues from across Canada. He is currently involved with research related to the development and use of plant resistance for disease control in cereals, studying the impact of cropping systems on disease development, and plant disease surveillance and risk assessment, and the development of practical integrated solutions for crop health and disease management. So whenever you're ready, Kelly, we can put your presentation up on the screen and you can take it away.
Yeah, if you can activate the share, it's
yep, absolutely.
Make me the presenter. Thank you.
And could you turn your camera on as well, if you don't mind?
Oh, sorry.
Oh yeah, no problem at all. There we go.
Sorry. Oh, okay. Oh, there we go. Great. And we'll put that in presentation mode, obviously. How does that look?
Looks good. Yeah, you're good to go. Take it away.
Great. I'll just sit up. So thanks very much to the Sask Ministry of Ag staff for inviting me to present to you today. It's always great to, even if it's virtually, to interact with colleagues and farmers and industry staff and so on in Saskatchewan. I was born in Humboldt. My dad farmed around Dalesford, south of St. Preux, and I spent most of my time in Saskatoon. I'm a Sutherlander, so I grew up in the Sutherland neighborhood. It's the west side of the east side, as I like to say. Anyways, what I'd like to give you is a bird's eye view of some things to think about in terms of the upcoming growing season, and if we have a return to maybe more average types of moisture conditions. So with that, I'll move into the next slide, and that's really just very quickly to acknowledge the staff that I've worked with across the Prairie region, including our multiple centers and farms in Saskatchewan. So the technical staff, scientific staff, producers that we've interacted with, and industry colleagues, producer groups, and so on. Our program has been very fortunate to have funding from a number of different agencies for a variety of different research projects. And last but not least, thank you, Ali, for contacting me and expressing some interest in me blathering on to you this morning. So hopefully it's not too much blathering on. So if we look at the last several years, one of the questions that I've gotten is, we've had really dry growing seasons, either prairie-wide or certainly in certain regions where things have been quite dry, especially in July, which is a key month for foliar disease development in cereals and fusarium head blight. Mid to late June is also important in terms of getting things starting to go and being prepared from the point of view of a pathogen to infect that developing crop, especially during that key grain filling period. So there's dry conditions. I don't see a lot of disease. We haven't had downgrading due to fusarium damaged kernels. We haven't had a lot of leaf spots. Maybe haven't seen any response to our fungicides that we've tried. Shouldn't that limit my risk in future years? And the answer is not necessarily. And one factor to keep in mind is that you might have had moisture earlier in the summer, or sufficient moisture to keep things a little more on the humid side right at the soil surface and maybe up a few inches. So there's enough moisture in some cases to facilitate pathogen development on old crop residues, spore production, and infection of the very lowest part of that plant canopy. And those infections, because of the dry conditions, remain there, and they may be at a very low level, a trace level, but they're still there, but they remain in those lower canopy plant tissues, and they really don't move up into the middle and upper canopy. So when you're out looking at your field in late July, early August, you're not seeing a lot of leaf disease on those key upper canopy leaves. The problem is those infections lower down maintain a level of inoculum that can be a factor in future years. One of the other considerations too is that dry conditions will slow down residue decomposition and inactivation or active colonization and decomposition of pathogen survival structures. So you might have a source of disease that remains for a longer period of time. And in the 80s, we had a pathologist, Dr. Alan Petrie at Saskatoon. I was a summer student there in 83, and Alan did a lot of interesting work with virulent blackleg. And what he found actually, and I'll use my pointer, is that in the Saskatoon region, the typical rotation that was recommended for blackleg was a 4-year rotation. Didn't have fungicides. All our varieties were ultra susceptible at that time. You go up into the Melfort area, northeastern Saskatchewan, the recommendation there was a 3-year rotation. And the difference was due to moisture. So in the Saskatoon region, drier conditions, slower decomposition of those lower stem bases, and especially the root pieces of the canola plant that carry the blackleg pathogen. So an example of impact of moisture in terms of residue decomposition. So as I mentioned, over the years, I've had a lot of questions from silage producers, especially where they're using barley, and they say, well, you know, we're a bit frustrated. We harvest our silage, we're removing almost all the residue, and we still have horrendous problems with leaf disease development in subsequent years when we go back to barley. And that's a situation where the farmer may need an on-farm source of forage or silage. So they're growing barley and barley and barley. More and more over the last 20 years, that has switched to corn, certainly here in Saskatchewan, but I get the same impression in Alberta, and same impression in Saskatchewan. So the thing I say is that have a look at those lower stem pieces that are still in the field. And you can see on this leaf here, sheath here is a big scald infection. There may be some net blotch infection. This is a picture that's showed a lot of foliar development of disease, but if you can imagine limited disease development, but you look down on those lower leaves that are dead now, the lower leaf sheaths, you're going to have net blotch infections. And basically, if we look at tan spot, net blotch in barley and tan spot in wheat, the fungus survives as these specialized small black fruiting structures called pseudothecia, and they form either, let's say in the 25 growing season, maybe the late summer, early fall, and then the pathogen either survives as those structures or as hyphae that are quiescent. And then in the next growing season, moisture conditions trigger the formation or further development of these structures. And that video showed you an example of the structures on tan spot. Producing these sacs full of spores. And when you have those structures imbibing moisture, they become very turgid, and they actually shoot those spores, those ascospores, out like little cannonballs, and those will fall on the plant. So you might say, well, geez, I don't have a lot of infected residue. I might see the odd leaf here and there in the field. But it's really not going to be a problem. Well, the thing to keep in mind, if you look at all of our leaf spots in cereals, so tan spot in wheat, the septoria complex in wheat, net blotch in barley, both forms, spot blotch, scald, the rust, leaf rust, stem rust, and stripe rust. My apologies for that. Basically, they're polycyclic diseases, and just to illustrate that, so you have old crop residues, maybe not a lot of them. It initiates, and you see the odd net blotch lesion on some of the lower leaf tissue. Keep in mind that those symptoms are producing the asexual stage of the net blotch fungus, very similar in tan spot. These are cigar-shaped spores that are dry, they're dispersed by wind, they're carried to adjacent healthy plants or up to the middle and upper part of the canopy. And if you have suitable moisture conditions, they germinate to produce a new crop of lesions, which produces a new crop of spores, which initiate more infections. So this life cycle in the case of net blotch and tan spot and septoria takes about a week to complete. Something like scald is a little likes colder conditions, so it's about 10 to 14 days to complete the life cycle of scald. What really drives this, and I compare it to putting your foot on the gas and tromping on that, is highly susceptible varieties that are being grown and favorable weather. So even though you might be on a relatively extended rotation, or you've had dry conditions, so you don't have a lot of symptom development in previous growing seasons, this cycling, especially on a susceptible variety and highly favorable weather, can mean that even a relatively small amount of disease earlier in the season can build quite rapidly, again, with highly susceptible varieties and favorable weather conditions. So if we look at the 5 riders of the prairie barley apocalypse, scald is an issue mostly in Alberta. And I love working with barley leaf spot diseases because they're mostly distinct, especially some of the major ones. So scald is mostly an issue in Alberta, sometimes maybe in that Medstead, Meadow Lake area, sort of northwestern Saskatchewan, you might see something there, especially with the susceptible variety. Net blotch, though, it tends to be prairie-wide, one of our more consistent problems. Spot form net blotch, maybe not as much of an issue. The problem is spot form net blotch is easily confused with spot blotch. So often what we need to do is take these leaf tissues into the lab, process them, and look for fungal fruiting structures in these lesions or coming out of the lesions in order to identify whether it's spot form net blotch or net form net blotch. I wouldn't worry too much about what it is. It's destroying leaf area and having an effect on that plant's capacity to capture light energy from the sun and turn it into carbohydrates. One thing that I'll just mention is that if you're out scouting, one thing to keep in mind is look at your fertility program, because chloride deficiency in barley will produce symptoms that are very similar to spot form net blotch and spot blotch. There's some things that you can look at, but typically with the leaf spot diseases, they're most severe in the lower, and then there's a gradient of reduced levels of disease as you go to the top part of the canopy. Very indicative of a stubble-borne disease that initiates infections in the lower canopy and then moves up. In contrast, chloride deficiency in these symptoms, you'll get symptoms throughout the whole canopy typically. We can also get sun damage. So if it's been cool, wet, all of a sudden you get sun coming in, hot, dry conditions, really sunny, sometimes that'll cause physiological leaf spotting. So you get localized areas of sun damage. And as you can imagine, with sun damage, most of the symptoms are confined to the upper canopy. Any of the leaf tissue that's shaded further down typically will not exhibit those symptoms. The other big issue and a big problem, because our tools to manage it are really not, to my way of thinking, sufficient. We're going to talk a bit about that later on, and maybe some tips to tweak your management program for that. Unfortunately, we've got a sixth rider, and that's bacterial leaf streak, and I'll touch on that a little later on. It's something that became established in the northern Great Plains of Minnesota, North Dakota, South Dakota, and so on in about 2007, and has become one of the main leaf disease issues for growers there, and we'll talk a bit more about that later. It's something that we've seen over the last 25 years, but mostly in our disease screening nurseries at Lacombe, not typically in the commercial barley fields that we survey or wheat fields that we'll help survey on occasion. But it's been something that we're starting to see more frequently over the last probably 5 to 10 years in commercial fields. In wheat, the main issues are the septoria complex, and you've got speckled leaf blotch here, and then leaf blotch or gloom blotch down here. My colleague Reem Akabuka Hadar at Lethbridge is, in addition to Maria Henriquez at Morden, our leaf disease experts, and Reem's finding some really interesting issues with the septoria complex, that it's not one pathogen. And actually one of the new things that she's discovered can be more of an issue actually in barley, which we normally don't tend to see a lot of septoria in barley. Tan spot's the other main leaf spotting issue. And in general, just a general comment, whether it's barley or wheat, your main yield robbers and factors that influence kernel weight, plumpness, thins, bushel weight or test weight are the leaf diseases, whether the leaf spots or the rust. Fusarium head blight might produce some yield loss, but the main factors are associated with grade reduction in the fusarium damaged kernels, and of course the production of dawn in that grain. So tan spot is also the same. So the causal agent there, Pyrenophora triticirripentis, also causes red smudge and germ. And that's not something that farmers like to see, or the pasta makers. So if you get a lot of red smudge on the current grain, it imparts pinkish flecks or reddish flecks in the pasta, and that's obviously a no-no. And if you look at the rust, leaf and stem rust have traditionally been our main issue, but thanks to the breeding efforts starting way back in the early 1900s, and especially activities since about the 1950s and 60s, ongoing surveillance activities from pathologists, especially my rusty colleagues in Morden and previously at Winnipeg, we stay on top of these pathogens. We identify shifts in virulence, and then we work with the breeders to incorporate more effective genes for resistance. So we've been able to stay on top of that. Stripe rust has been an issue mostly in the Western Prairie region under cooler conditions, but within the last probably 15 to 20 years, there's been the emergence of a warmer temperature biotype in the States. So now in Oklahoma, Texas to that Nebraska corridor, you can get a lot of stripe rust. But traditionally, stripe rust was more of an issue in southern Alberta, maybe some parts of southwestern Saskatchewan. So, and then again, fusarium head blight, and then unfortunately, we've also got bacterial leaf streak in wheat. And there are a couple of different types of the Xanthomonas translucens bacterium. Some can attack both wheat and barley, some can just attack barley alone. Just a quick plug for the Prairie Crop Disease Monitoring Network, and this is an example of what I'll send Ali to distribute to you in addition to a PDF version of the presentation. It just shows you some of the information that's available, and we've worked quite hard with people like Aliriza Akavan in Saskatchewan, Mike Harding in Alberta, and David Kaminsky in Manitoba. So we pulled together a suite of disease info cards that provide a bit of an overview of the disease, the symptoms that occur, and then we've referenced those to some pictures. One of the techs I worked with yesterday said, Kelly, you've got too many arrows on this, so we're looking at that. My apologies. And then we have a diagrammatic representation of some of the main disease management strategies and some caveats there and so on. So if we look at risk factors, I think that's an important thing to consider in terms of what you might expect. If you can look at these risk factors, relate it to your farm or the farm, a client that you're working with, a farmer client, and that can give you a pretty good idea of what issues may or may not occur and the factors that drive those. The other thing is that if you're a farmer or you're advising a farmer, when you look at disease management, you're really looking at manipulating one or ideally multiple factors that are related to the host, the pathogen, and the environment. So if we look at the host, a susceptible variety. So that is certainly the plant can't prevent attack and the damage that an infection will cause or that a pathogen will cause. Increased yield potential. If you're driving for high yields, you're pumping the nitrogen fertilizer, you're going to get a dense crop canopy, which tends to hold the moisture in for a longer period of time, and that actually might promote greater disease development. The fertility status, and I'm talking about the extremes, so deficiencies and bona fide deficiencies or excess nutrients can sometimes cause phytotoxicity. So keep that in mind when you're looking at your fertility program. You want to have balanced fertility. The pathogen itself, you're targeting directly with crop rotation. So a tighter rotation obviously is going to be result in increased risk. Pathogen virulence, so can that pathogen attack the variety that I'm growing? Do the genetics in that variety, are they still effective? And pathogens are not static, whether you're looking at cereal diseases, things like blackleg and clubroot and canola and so on, they will adapt to the varieties and the genetics that the breeders and pathologists have incorporated into those varieties. One thing that we often forget about is seed health. And things like Fusarium graminearum, the spot blotch fungus, have significant impacts on seed germination, seedling growth, and stand establishment. So definitely get a fungal disease screen on the seed you're intending on planting. And often you really need high levels of infection of graminearum or the spot blotch fungus. I'm talking not 5, not 1, I'm talking 10 or 25% or higher seed infection. And you can get that routinely in some areas in the prairies. And then field location is something we often forget about. So if you've had an issue in 2025 with, let's say, Fusarium head blight, you're up in that northeastern part of Saskatchewan, that field, if you plant immediately adjacent to it with the same crop variety, can serve as a source of disease. There can be some movement, short distance movement from one field to an immediately adjacent field. The environment, the mother of all risk factors. And rainfall and RH are the key parameters. Temperatures, I would say, in the extremes. So your temperatures, if they're in the range of 15 to 25, Those are conditions that favor disease development for a wide range of disease issues. As you get closer to 20 and 25, yes, that's going to drive things a bit more, but it's really the extremes. So low temperatures below 10 or above 30 and above daily highs, those will slow down things in terms of disease development. Irrigation may increase your risk. And then the crop canopy environment, and that kind of relates to yield potential, your amount of fertility, your seeding rate and row spacing. I've got question marks there. Often they're attributed increased seeding rates or narrow row spacings might increase risk. That's not necessarily the case. So if we look at the prairies as a whole, and I pulled this off of Stats Canada yesterday, we're looking at Alberta, Saskatchewan, and Manitoba. We're looking at the percentage of acres planted to barley, canola, oats, field peas, and wheat, all types. And I just tried to keep things consistent so we don't have chickpeas or lentils, just because we don't have a lot of that in Alberta. So the key thing is I want you to look at the amount of canola and the amount of wheat and the percentage of acres. And really, if you combine these two crops in Alberta, 72% of the acres are planted to either of these two crops, 83 in Saskatchewan and 87 in Manitoba. So what that means is that we're looking at a canola wheat rotation. In some areas, maybe in Saskatchewan, that would be a wheat lentil or a wheat chickpea rotation, but maybe you're extending that because of the Phanomyces or the Ascochyta microspherula complex. And then in some areas where they love to grow malting barley, it might be a malting barley canola rotation. So if we look at crop rotation, it's a major disease risk factor, but only for residue and or soil-borne pathogens. So the field itself, what is in that field, what have you grown previously, what has occurred previously will impact your risk in future growing seasons. So you need at least typically a 2-year rotation with a non-host crop. And unless you have that, you're going to see an increased risk. Basically, you're going to build the amount of infected crop residues. You're going to build the number of pathogen survival structures either on the soil surface or in the soil itself. And unfortunately, with a tighter rotation and the same varieties, you're going to select for more virulent races that overcome the genetics for resistance that have been incorporated in those cereal varieties, or in the case of canola, blackleg, and clubroot. Poor weed control and volunteer control. We often don't think about this, and it's something that I've chatted a lot with Brianne Tideman here at Lacombe, a young weed scientist, but keep that in mind, and that can cancel your rotational benefit. The other thing that it might do is if you're using fungicides, and a lot of the fungicides that we have, the actives tend to be fairly common. They mix things up a bit between what you spray in your wheat, your barley, your field peas, your canola, your lentils, and so on. So keep that in mind. And what you might see is that facilitates selection for fungicide resistance. And we'll talk a bit more about that later. And then the other aspect here with rotations is it can be quite challenging to manage diseases that have multiple hosts. So Fusarium graminearum can infect wheat, barley, canola, rye, triticale, corn or maize, sorghum, and so on and so forth. Same thing with sclerotinia and canola. It affects all of the broadleaf weeds and crops that we have in the prairie region. So those multiple hosts may impact the effectiveness of rotation. So if we look at crop rotation, it really needs to be considered along with other factors that influence a farmer's decisions. And I have always kept a few things in mind that friends of my dad or others have told me over the years, and being respectful to farmers and so on, that, I don't want to get up here and preach, thou shall rotate at least 2 years between host crops, so on and so forth, because there are many factors that influence a farmer's rotational decisions. Commodity prices and stable markets would be the mother of all factors. What is going to make me money? What is going to pay my bills, etc. On-farm feed and forage needs is another factor you need to be mindful and respectful of. And last but not least is, we often don't think about this, the familiarity with the crop. I know how to grow wheat and canola well. I have the tools largely, maybe with Fusarium head blight, they need some tweaking to manage those risks. So that's something to keep in mind. So my grandfather, I never did meet him. He was homesteaded in the Dalesford area and was in World War I. But climbing around as a kid on the farm, I found this textbook that he had, Farm Economy, 12 Courses in Agriculture. And one of the chapters had lots of information. And the more things changed, the more they stayed the same. So old information, if you're a younger person, I'm getting up there, I'm 63, don't pass on it, because some of that old information is foundational in nature and is still relevant nowadays. So back then, the call was rotations need to be based on crops that are profitable for the producer. Otherwise, the benefit in terms of disease management is rendered useless. That was 1921. I would argue very strongly that the same idea holds true for farmers in 2026. So one thing that I always like to do is talk about the scenario, what if I have to look at that tighter rotation? What can I do to mitigate that? Well, the farmer and the consultant advising them needs to understand and manage that risk. So there's an increased risk of disease source buildup. There's potential for shifts in pathogen virulence and fungicide sensitivity, especially if you're using fungicides at least 2 or more times in one growing season and then under short rotations between years. Be extra vigilant, monitor, monitor, monitor. I can't emphasize this enough. That information is critical in terms of developing a plan to mitigate some of that risk. So check the seed you're intending on planting, scout the fields during the growing season, whether that's you or a professional consultant you're working with, and test the harvested grain that you're looking at. So very important. And my colleague, he's got to be in his 80s now, Ian Evans, the famous, world famous Ian Evans, he introduced me to the NIMBY syndrome, not in my backyard. And in the late 90s and early 2000s, farmers in Alberta were looking at over the Red River Valley in Manitoba, western Manitoba, southeastern Saskatchewan, and they were seeing increasing problems with Fusarium head blight caused by Fusarium graminearum. And I can tell you, this syndrome was that's a Manitoba or eastern Saskatchewan problem, never become a problem in Alberta. Fast forward to 2026, yeah, absolutely a problem in Alberta. Utilize all relevant tools, and really you're just trying to compensate for a lack of rotation. Now, from my point of view, as a researcher, we need to look at addressing these risks and coming up with strategies to try and mitigate them. And often my view is these are short to medium term solutions, because ultimately highly effective strategies may be very difficult to develop, and these strategies may not provide durable management of those issues. So eventually the disease issues become so bad that they have managed the farmer. And we saw this in Saskatchewan, especially that northeastern part of the province, but elsewhere too, as we moved from the early to mid to late 80s. We had susceptible varieties, and basically farmers had to adopt a 3 to 4 year rotation. Otherwise, they couldn't grow canola without having significant blackleg issues. Nowadays, resistance has mitigated that. And the other one is Aphanomyces in field peas, and that's knocked a lot of producers either out of growing field peas, or they're going to a 6 or now 8 year rotation, which is quite unheard of. So let's look at options. So we've interacted a lot with crop specialists when the province had them, unlike Saskatchewan. Often they would, especially southern Alberta, say we have got clients with horrendous leaf disease problems. How do we manage this in a silage production system? So we did some work at Lacombe over a 9-year period, and we looked at different strategies. So the lowest diversity was the same barley variety, sundry barley, grown 3 years in a row, and we had 3 rotational cycles. So basically this was 9 years in a row by the time we got to 2016. So you can see extremely high levels of leaf disease on the penultimate leaf. So you're looking at a significant reduction in biomass and tonnage, whether it's on a wet or a dry weight basis. We looked at variety rotation, so changing varieties each year. It helped a bit. lower it down a bit. We then looked at barley variety mixtures, but within that 3-year sequence, the 3 components stayed the same. So variety 1, 2, and 3 were grown in each of those 3 years. We also looked at mixing things up. So each year we grew different varieties. So year 1, variety 1, 2, and 3. Year 2, variety 4, 5, and 6, and so on. We also looked at intercropping. So you can see a downward trend. This tended to give us where we used a mixture and changed the components each year. We introduced wheat and spring triticale, and that resulted in a reduction. But if we used the same varieties, it wasn't as great as we had hoped. If you looked at intercropping with spring triticale, oat, and barley, we could bring that down, maybe not as far as this one here. And then we had winter triticale, and that was planted in the spring as a mix with 100 seeds per meter squared of oat, barley, and triticale. So this remained in a vegetative stage. And actually what we found is that the intercrop with winter triticale and different varieties each year gave us our lowest level of disease. And that understory of vegetative material from that winter triticale planted in the spring helped to interfere with spore dispersal from the residue plus the lower leaf infections. Now, what about tonnage? That's the most important parameter. It's a dry weight basis. And again, and as we increased the amount of diversity, we increased our tonnage. And really, it was the intercrop with spring triticale and different varieties that gave us our highest yields. And here, the spring triticale would be producing grain. So you get, and at the soft dough stage, you'd have some addition of that grain material to the overall tonnage. So definitely a strategy to look at. We worked with Tim McAllister on quality of that for feedlot cattle, and it was perfectly fine, that mixture of barley, oat, and triticale. So if we look at crop variety, that's an excellent tool to manage and probably one of the more important ones. So susceptible varieties, as I said earlier, and I won't belabor the point, simply can't prevent infection, or they can't restrict the development of that pathogen in that plant tissue. Resistant varieties, and resistance isn't necessarily immunity, although with some of the highly effective rust resistance genes, you can essentially have no rust development there. But really with the resistant variety, it restricts that spore's ability to infect the plant initially, and in some cases, how fast that pathogen grows through that plant tissue. Now, again, shifts in pathogen virulence can overcome these resistance genes. So the genes themselves in the plant don't change. It's the pathogen population that's adapting to that genetics. Now, resistant varieties may preclude the need for a fungicide, but the problem is you're not faced with one single issue. So stripe rust. You might have tan spot and septoria. So you need a complete suite of disease resistance in varieties, and that's a difficult animal to do, or to a process to do. And then other plant characteristics. So lodging, viney growth tend to increase disease simply because you get a very humid microenvironment in that lodged or viney crop growth habit, and that can increase your risk. So you might look at this and think, well, this is a scald resistant variety, very limited disease development. And here's a scald susceptible, quite extensive scald development, maybe in that moderate range typically. But what you're looking at is a trial we did in, this going back a little ways, 99, 2000, 2001, where we looked at barley variety rotation. So we had Casota, and where we grew it 3 years in a row, we shifted the pathogen population in those plots. So the pathogen, the scald pathogen, overcame the single gene for scald resistance that the breeder had incorporated. Now, I'm just going to talk briefly about bacterial leaf streak. It's an emerging issue, and Ali said, you know, please touch on that. So one of the key diagnostic features of bacterial leaf streak is sort of these greasy, water-soaked lesions initially, and then if you have humidity, the bacterial cells will divide and you get millions or billions of bacterial cells, and you get oozing of masses of bacterial cells out of these long streaks and lesions. Eventually, these lesions, the plant tissue will die, and it may turn a darker color. But the water-soaked, greasy appearance of the lesions, the oozing, are very diagnostic symptoms. The other thing is if you go out in the morning and there's dew and you rub your fingers along that leaf, or if it's on the head, the chaff phase or black chaff phase of it, it's extremely slippery. So keep that in mind. So we have some information on the Prairie Crop Disease Monitoring Network. I'm not going to go into a lot of detail. This will be one of the PDFs that I send to Ali that she'll distribute later. So we've got a description of the disease, key characteristics associated with the symptoms. We then refer to those symptoms on the next page where we have typical symptoms. And then we provide a diagrammatic overview of the key management strategies. So the challenge with bacterial leaf streak is there are no in-crop treatments, no foliar products that we can apply, no seed treatments. They have looked at this extensively in the northern Great Plains of the US and unfortunately have not been able to find anything that is effective. And fungicides are focused on fungi. They do not have any effect whatsoever in terms of bacterial leaf streak. So the main focus is field scouting, seed source. It's highly seed-borne, so work with your seed testing laboratory to give you an indication of level, and then variety. And that's something that we've been working on over the last 3 years in conjunction with Randy Kuchar, as well as expanding some of the barley cluster work that we do in terms of disease-resistant screening. So to give you an example, here's the 2-row malting co-op trial at Lacombe in 2024. Here are the candidate lines that the breeders are proposing. Here's our minimum rating on July 24th. Here's our maximum rating. Here's our average rating. So we've got a range there from things that are moderately resistant to things that are on the susceptible side, and some of our checks. So you can see that the great thing is that we have some genetics there. We expanded this trial in 2025 to include some BLS checks. I want to point out that CDC Bold and Copper, some varieties from Aaron Beatty's program, they're on that more intermediate to moderately resistant side. Older varieties, a 6-row variety Mohegan from the program here in Lacombe with the province, about 8, and then Mavericks sort of intermediate. And then our co-op trials. And you can see some of the more common varieties like Synergy. It's actually not too bad, but again, focus on these blue lines. And we have some genetics out there that the breeders are working with. And these are lines that are close to being introduced as varieties. So this is the last part of the variety development process as part of the Prairie Recommending Committee in oat and barley. I'm looking at barley here, but if you talk to Randy, and we've done work with Randy and supported him, same thing with wheat. So here's some of the, we expanded our screening nursery in 2025. Unfortunately, with Lacombe being shut down, this is something that will be dropped, unfortunately. Perhaps my colleague James Tucker in Manitoba can pick it up. But if we look at the programs, here's some of the average ratings of the breeding material that they sent us for screening, some of the minimum ratings. So man, oh man, there's some exciting things here in terms of host resistance. So that's one thing that I've devoted my career to, is strong support of the breeding programs across the Prairie region. So I'm really gonna miss that. So again, there's some light at the end of the tunnel, and I think my colleagues will continue with this work here in Lacombe, Aaron Beatty in Saskatoon, and Anna Badea and James Tucker in Brandon. So often over the years, talking with breeders, typically in the bar at the variety registration meetings, pathologists tend to be pretty tough on their lines. Sometimes they're pretty crappy as far as disease susceptibility, and we're not shy to let them know that. So the comment is, well, we've got fungicides. We can spray fungicides. It'll take care of our problem. We won't need to worry. Maybe, maybe not. So it's just a slide to illustrate an example of that. So yeah, no worries. I'll just hit it real good with fungicide. Now, fungicides are great. They're an important tool available to producers, but you need to be mindful of the role they play and when they will not be a strategy that you want to look at. And here's some trials as part of the wheat cluster project that we had from 2018 to 22, AAC Brandon wheat, and this was at Beaver Lodge, 2018 through 22, and we had the first two bars are leaf disease, no fungicide, leaf disease level with fungicide. So you can see in 2018, Greg Semich, our key collaborator there, had about 25% leaf area affected on the penultimate leaf at about the late milk, early dough stage. We could reduce that significantly with fungicide. If we looked at yield with no fungicide and fungicide applied, we saw a significant reduction. So our checks, we had about 25% leaf area affected, but in all the other years where our levels of leaf disease at the end of the growing season, late July, early August, were well below 10%. And in all cases, we did not see a significant response from fungicide. And it was Procero XTR applied at that anthesis timing. Very similar picture with some of the barley work that we've done over the last 30 years. One thing I want to talk about are the cereal rusts, and these are issues, especially stripe rust, that I'm more familiar with here in Alberta, that have snuck up and really hit producers hard over the last 25 years. Typically in commercial fields, the farmer or consultant says, well, geez, there's rapid and sudden development of damaging levels of rust. And by the time I recognize that problem, it's too late to spray a fungicide and to get the most out of that fungicide in terms of application. So in general, cereal rust typically do not overwinter on the prairies, especially things like stem rust, maybe to a certain extent leaf rust, but especially stripe rust. They tend not to it's mostly stripe rust, little less for leaf rust, and typically with stem rust, that's not the case. So they survive on cereal and grass hosts in the United States, and we'll talk a bit about source locations in a minute. However, stripe rust, and Randy Kuchers has seen this, and we've seen it in work that we've done here in Lacombe with Alberta Ag, in southern Alberta, central Alberta. It can move from the spring crop the previous summer onto the winter wheat crop that's been planted in September. If it's a mild winter, that stripe rust fungus will survive, and once that winter wheat plant starts regrowing in April, that stripe rust fungus will start regrowing. And here's an example of that at Olds, Alberta. With Alberta Ag, I was down with some colleagues, Pathologist, Kik Wan Shi and others, and Krishan Kumar in mid-April. And we saw extensive stripe rust starting to develop. So that would contribute a significant amount of inoculum for that field. But rusts are a communal disease, so that would be a source of disease for adjacent spring wheat crops and even barley crops potentially. So typically, spores of the stripe rust fungus are blown northward from the US, so that Texas, Oklahoma, Kansas, Nebraska, up into the central to eastern prairie region, and Oregon, Washington State, and northwestern Idaho into southern Alberta and southwestern Saskatchewan. So if we look at stripe rust, fungicides in a susceptible variety are highly effective. So if we look at a trial that we did with Randy Kucher and Keiko Nabatani, a master's student, this is at Lacombe, Bellatrix is susceptible, no fungicide, a full rate of twin line in the fall or in the spring or a dual. You can see that fall application really didn't have much effect. It was really only once we put that fungicide on the flag leaf stage in the spring, protection of the upper canopy leaves, I might add. The dual really didn't provide much additional benefit. Radiant is a moderately resistant variety, at least at that time in our area. The stripe rust pathogen has adapted to the genetics. So you can see the blue bars here are simply the role of host genetics. And you can see went from 100% severity down to about 18, maybe 17, 18%. Fall fungicide didn't do a lot. And it was really where we put that product on in the spring. What about yield? Well, you can see with Bellatrix, a little bit of a bump up with the fall, but not statistically significant. It was where we put that fungicide on, either singly in the spring or with a dual fall and spring. And you can see no additional benefit to that dual. With Radiant, flat response. So the genetics were doing the heavy lifting here. And just to emphasize the role of sudden appearance. So, you go back, and I think it's important to keep in mind history. Don't, if you don't remember your history, you're bound to repeat it, as the saying goes. So I got called out by a seed grower in August, early August. He had a problem in his wheat. I think there were some fertility issues and maybe a late herbicide application problem, but there was a moderate to severe stripe rust problem, and our pants and boots were covered in yellowish orange urea spores. The other case was in July 2026 in Kelly Cooley with the municipality of Pincher Creek. Had a farmer who said, I drove by the field on a Friday. It looked good from the road, 60-mile-an-hour field inspection. I drove by it on Monday, and it was starting to look yellowish orange. And these spores, Kelly went out, the producer decided to silage the crop. It was originally meant for grain, and their silage harvester was absolutely covered in masses of the Rita spores. So sudden appearance. So depending on when that disease appears, this is some work out of Australia. And if you ever get the opportunity to listen to Steven Simpfendorfer and Ellie, I would recommend him as a potential webinar presenter. Great fellow, loves his beer. I visited with him twice over the years in Australia. Simpy, as his nickname is. And he pointed out that if you get rust, stripe rust, entering the crop at around the first node stage, growth stage 31, and highly susceptible or susceptible varieties, even in moderately susceptible, your yield losses range from around 55 to 85% or more. Moderately resistant, and that's the highest level of resistance they had, 25%. But as the arrival date becomes later, you can see the level of yield loss for all of these varieties declines rapidly. So if it comes early, and so you might say, well, yeah, Kelly, that's Australian data. What about Western Canada? Well, if we look at work done in Lacombe and focus on the red box here, in general, yield losses of 4 to 22% occurred. So 22 would be the more susceptible, 4% would be the more resistant when the rust appeared at late heading versus about 11% to 91% yield loss when that disease appeared around the emergence of the penultimate leaf, I think around growth stage 37. Super, I'll move through this, I'll go through this quickly. The main point of this here, Symphia again, is that they had huge issues in 2022. They had a more of a range of susceptible varieties, and they had really wet conditions that precluded them getting in to spray weeds, but in the case of diseases, fungicide application. And that large amount of inoculum was also a concern from the point of view of pathologists in relation to potentially seeing shifts in the virulence of the stripe rust pathogen population. So some other information, and I'll break this out for you in the slide deck. But I spent a fair bit of time with Mark McLean in his lab at Horsham, north of Melbourne, in 2008 and 2012. He retired from pathology and decided to go farming and sheep production. But he was telling me in November, they were not able to get into the crop from late June to late November to spray anything. They had aerial applicators going. Problem is the number of applicators was greatly reduced, prior to maybe 30 or 40 years ago. So the wait for the plane was about 2 to 3 weeks. So fungicides are an important tool, but if you can get it, only if you can get into the crop to spray them. So just quickly, a plug for the Prairie Crop Disease Monitoring Network. So during each of the summers of 2019 to 25, we issued 7 to 10 cereal rust risk forecasts. These were based on the occurrence of wind events from key prairie locations with running summaries of the total. So these were from the source areas in the States. We looked at rust development in these source areas, so Texas to Nebraska and Pacific Northwest, and then looked at prairie weather conditions and crop development, and then issued rust risk assessments and recommendations not to spray, but for timely in-crop scouting. So you could look at getting ahead of this disease with that heads up, and that's the purpose of these risks. Just very quickly, if we look at the Pacific Northwest, most of the trajectories end up in Alberta and Western Saskatchewan, and that trip from Pullman, Washington into the Lethbridge area takes less than half a day. if we look at that Kansas to Nebraska corridor, same thing would apply for Texas and Oklahoma. Most of the trajectories end up from central Saskatchewan into Manitoba. So again, what we do is we look at rust development, we look at wind trajectories, we look at prairie weather conditions and crop development, and we issue, a risk forecast. So herbicide timing, or fungicide application, timing is a critical factor. We've done a lot of work at Lacombe, especially on barley. We did a little bit of preliminary work on wheat, and all of that indicated that that herbicide timing is not the right target. So here's Checkno fungicide, extensive scald. Here's Tilt, half rate at the 2 to 3 leaf stage. Really didn't do much in terms of disease management. And here's where we put that product on to those key upper canopy leaves. Work done by Sherry Stridehorst and colleagues, master student Manur Asef. And basically, herbicide timing and PGR timing, there was no benefit. It was only where you put that fungicide on a flag leaf and head timing in wheat where you saw a significant increase in yield that would likely cover your input costs. So just to emphasize this, and I visited with Nick Poole in Australia. We had him over in 2013 to speak at some meetings. He refers to these upper canopy leaves as being the money leaves. So in wheat, it's the flag and penultimate leaf that are most key, and the head in barley. The flag leaf tends to be quite small in a lot of varieties. It's the penultimate leaf, the third leaf down, the flag leaf sheath, and the head itself. So you want to protect those money leaves. That's your key target for foliar fungicide applications. Just being cautious about fungicides, another caveat. Europe is the basket case in terms of fungicide resistance over the last 30 to 40 years. I look at Australia as the canary in the coal mine. And if you look at net blotch, it has a net form resistance to the triazoles. It has resistance to the SDHI category, which is a relatively new group here in the prairies, and resistance, or at least reduced sensitivity to the strobilurin class. Nick gave a webinar in 2024, and he said for the first time at the FAR Australia Research Centre, the net form of net blotch was poorly controlled in RGT Planet. It's a European variety. We're seeing that introduced here, highly susceptible to net blotch. And even with 4 units of fungicide, so a seed treatment, start of stem elongation, flag leaf emergence, and head emergence, they could not control net blotch. We are seeing shifts in the triazole class, especially propiconazole, and we just had a student finish up and defend her thesis last week or the week before, and there's some indication of potential shifts in sensitivity to the spot blotch pathogen, especially in the case of the SDHI class. So with FHB, we'll go through this quickly just to stay on time. I just want to emphasize that, you know, I like to keep an ear to the ground. So I talked to Faye Dolken-Bouchard over the years when she was the provincial pathologist, Randy Kuchar. And in that northeastern Saskatchewan, growers are doing what the experts, and I use quotation marks, are saying: grow a resistant variety, avoid host-on-host rotations, and use a fungicide. But even when they do all 3 of these, when the weather is favorable and graminearm is well established on residues, they're still getting significant yield, but more importantly, grade losses and dawn issues. So I want to talk a bit about tweaking things, and we'll go through this quickly. So if you look at, in Alberta, the Guide to Crop Protection is called the Blue Book, and I should have changed this to 25, but it doesn't matter. So in wheat, the start of the window is 75% head emergence. In barley, it's 70% head emergence. So what does that mean? Well, it means that potentially in wheat, if you go at the early part of that window, 25% of those heads are not getting any fungicide at all, and you need to get that fungicide directly on that head tissue. And in barley, it's even worse, potentially 30%. If we look at the recommendations in 2024, at the time in wheat, it was at the start of anthesis. So roughly about 3 to 4 days after head emergence, you get yellow anthers in the middle part of the head. That was the key target. Anything later than that was too late. In barley, it was full head emergence, and anything later than that was too late. If we look at 2024, and these are graphics from Andrew Frischkopf and an NDSU crop and pest report, excellent resource, I might add, so subscribe to that. All the work in the US and Canada is now indicating that we can actually go a fair bit later in terms of fungicide and not compromise our control. Just quickly, if you look at infection in FHB, it occurs from head emergence through to the start of senescence, wide window. Makes it difficult to control. So if the infection occurs early, so within 5 to maybe 15 days of anthesis, that's when you get the most severe development of FDKs and the most severe downgrading. But infections can occur later. And if they occur later, you may not have that FDK. The wheat and the barley, nice and bright, normal looking, but they're still contaminated with DON well above the acceptable level. So let's look at timing. Here's some work out of the US coordinated by Pierce Paul at Ohio State. Here's 50% head emergence. Here's the ideal timing, traditionally, 50% early anthesis. 50% of the heads. Here's 5 to 7 days after early anthesis. And you can look at both fungicide actives. The trend was for improved control, still under or at 50% or under. If you look at some excellent work done by Gursahib Singh and Randy in Durham wheat, again, here's head emerged. This is the ideal timing in terms of fungicide. So you get bump reduction in DON, anthesis halfway, anthesis complete. So you can see even as late as growth stage 69, we're getting good control and we're not compromising that. And we had similar patterns with some work we did, but it was a period when we had dry summers. So from a pathologist point of view, it was a bit disappointing in terms of the amount of disease. We looked at 3 to 4 days after full head emergence, so start of anthesis, ideal timing, and we pushed the envelope. We looked at 7 to 10 days after that, so quite late. And you can see no fungicide, early, late single, or the dual. And this was a year and at Melfort, not a lot of DON. We had much more DON in 2019, so above 1.8, so knocks it out of the malting market, could reduce that to under 1 part per million with early, a little bit better control with the late, again, keeping in mind that's quite late. And actually we're seeing some impact of dual. And I would emphasize, I think you're starting to see some utilization of dual post-head emergence applications in the States to try and address FHB. Not on the label, I'm not advocating it. So how do we pull all this together? So if we look at that herbicide timing, it's not the right stage for leaf spots. It can be for rust, stripe rust, and leaf rust, and so on. I would recommend seed treatment. A lot of the actives actually move with the water transpiration stream into the first and second and third true leaf. So that might give you some peace of mind to try and limit things. But if you're seeing a lot of early season disease, you're out scouting for weeds, don't spray at that time. Your focus is going to be on applying a fungicide at flag leaf emergence or perhaps a little bit before that. And if you don't have any leaf disease and your primary concern is FHB, so the crop's clean until, let's say, a flag leaf and beyond, that's where your head emergence. But in reality, you're dealing with leaf diseases and concerns regarding FHB. So depending on the year, you may need to look at a pre-head emergence and a post-head emergence fungicide application. Just to emphasize delaying, so this is some work we did and a very similar picture with the similar trials with AAC Brandon and wheat. I just want to emphasize Lacombe in 2019. Here's the level of leaf disease. So we had around 57% where we put a fungicide on at the anthesis timing, so post-head emergence. If we delayed, we had a 7% reduction in yield. So in this summer, that pre-head emergence, so probably at penultimate or flag leaf, we didn't have that in this trial, would have been an important target to hit, and then to top that up again post-head emergence. So why do you see the difference between rust and the leaf spots? So you look at leaf spots and fusarium head blight. The 10 spots, septoria complex, net blotch, spot blotch, scald, depending on wheat and barley. They're all necrotrophic pathogens. They produce either toxins or enzymes or a combination thereof, and they kill plant cell tissue and they feed on the cell contents. So if you can imagine spraying a fungicide with well-established leaf spot infections, that fungicide will not eradicate that pathogen. That's a myth. It will struggle. It might suppress it for 2 weeks, and then that fungus becomes active again and starts producing spores. So the normal water conducting pathways are no longer active, and that may be one reason why eradicating a well-established infection is very difficult for the leaf spots. In contrast, cereal rusts and mildews are biotrophic. So they don't kill the plant initially. They produce these haustoria, which are specialized feeding structures that penetrate into the cells. They derive nutrients. So the normal water conducting tissue is still active. So that fungicide likely has a much better ability to penetrate into those rust infections and eradicate the pathogen. So just want to pull it all together in terms of FHB forecast. So what my recommendation is, start following the forecast around flag leaf emergence, and then booting, head emergence, and into the ideal timing. And if it's a low risk from that period, start a flag leaf, don't spray there. It likely means that you don't need to put a fungicide on for FHB, but keep in mind your leaf disease risk. If the risk is high, starting from flag leaf, so you get moisture conditions that are facilitating spore production. So the fungus is going to be in the air and it's going to be present at a moderate to high level as that head emergence occurs. Here, you might want to look at applying as soon after full head emergence as possible. The more difficult one is if it transitions from low to high. And I would recommend looking at your client, looking at their past experience with FHB. Have they had issues? For what it's worth, look at the forecast for the next 2 weeks. And this is where maybe application at the mid to latter part of that application window will be important. And then if it transitions from high to low, this is a real challenge. And again, I would probably recommend looking at the risk aversion level of the forecast, the 2-week weather forecast, I should say, and use that as a guide for the farmer in terms of whether to spray or not. For pulling it all together, combine fungicide applications with other strategies, no matter if you're looking at leaf spots or FHB, so careful seed choice and treatment. Try to use 2 years, recognizing the problems with that and the other factors that are much more important in terms of a producer's rotation. Use the most resistant variety available. Use residue management. Chop the residue and spread it as finely as possible and as wide as possible. That facilitates decomposition and a more rapid disappearance of a source of disease. Monitor harvested grain and seed intended for planting. So on, fungicide application can help. It's important to account for early to mid-season leaf disease development. That will dictate whether you're looking at a single or a dual application and when you put those on. So if you don't see symptoms until the flag leaf stage and beyond, that's where that post-head emergence is going to be a key target. If you have lots of leaf disease developing at tillering, stem elongation, and starting into upper canopy leaf emergence, that's where you're going to have to go pre-head emergence, either at flag leaf or slightly before, and then come back in and top that protection up with a post-head emergence application. Optimal timing for FDK suppression and dawn for FHB will vary depending on weather conditions and aerial spore load. So use the weather-based forecasting systems to help guide it. We're almost done. So utilize the latest in fungicide application technology. Tom Wolf is the go-to on this. So man, oh man, you know, the dual nozzle setup for FHB, the dual angled nozzle setup and so on. Ensure a uniform target. Uniform stand establishment and head emergence are critical, if possible. Mother Nature may intervene. Seed and seedling disease issues may intervene. It might result in a more variable crop. And this may be an area where in the States, they'll go in at the start of that window, maybe even a little early, because they've got variable head emergence. The main stems are emerging, the tillers are not. And then they'll go back in after full head emergence in that crop to provide protection to those late emerging heads. So this is where the dual might come into play, and if you look at the labels, there might be some options there. Direct application of that fungicide to the head tissue is critical. Focus more on the mid to latter part of that label window to get the best level of suppression of DON and fusarium damaged kernels. Ultimately, long-term, improving our ability to manage DON and FDKs in terms of fungicides may require a change in mindset, regulations, and chemistry. So the addition of later or dual post-head emergence applications need to consider pre-harvest intervals, maximum residue limits, and so on. And then, of course, for the farmer, the economics of that fungicide and the cost of application and chemical, and perhaps the introduction of actives. And some of the SDHIs might actually persist for a longer period of time. So with that, I'll end there. Here's my email address until probably late May or June. Here's my Twitter handle. I'll still have that. And with that, I'd be happy to answer any questions, and I will stop sharing my screen.
Okay, great. Thanks for that awesome presentation, Kelly. So the first question, when you mention suitable moisture for disease development, what do you consider suitable moisture, and will heavy dews be enough for disease development?
That's an excellent question, and it's important to look at the pathogens you're dealing with. So for things that are wind dispersed, like tan spot, Canidia, net blotch, or spot blotch. Those don't require splashing rainfall. The Septoria complex produces pycnidia and pycnidiospores. Scald produces masses of spores. Same thing with bacterial leaf streak pathogen. They require really frequent rainfall to disperse those spores from the lesions onto adjacent plants or further up into the middle and upper part of the canopy. So definitely heavy dews can facilitate development of things like tan spot and tan spot net blotch and the rust. There's a critical point where you need moisture to occur. You'll need more frequent rainfall and leaf wetness for some of these other issues, but those will also facilitate rust and tan spot and net blotch development. One thing that I would recommend, and it was an experience I had visiting a grower about FHB, a seed grower, but he wanted me to come out and help him decide whether to spray for sclerotinia or not. And we were out in the field about 10 a.m. in the morning, and we went to several canola fields just south of Red Deer, and they were bone dry. Soil surface was bone dry, crop canopy was bone dry. Those are absolutely not conditions that favor disease development, whether it's sclerotinia in canola or the diseases we've discussed in cereals. Conversely, if you go into that field later on in the afternoon, about 2 or 4, and that soil surface is wet, your boots are muddy, your pants are wet up to the crotch, the crop canopy is wet, those would be ideal conditions. So I say send the kids out, if you've got teenagers or kids, tweens, send them out in the quad, get them to walk through the crop and look at their pants. So Keith Gabbert, I was telling him about this, and he coined the term wet pants test. If you're seeing that moisture persisting in that crop canopy well into late afternoon, that is a clear and simple indication that you've got weather conditions that favor disease development.
Yeah, great answer. Thank you. The wet pants analogy is certainly applicable. I know the Canola Council has some promotes the same information. So, next question is: can tan spot look similar to a zinc deficiency? 2 years ago, this agrologist had very wet field conditions early in the season and saw high amounts of, in quotation marks, leaf disease at 3 to 4. Leaf stage in cereals, but could it be possible that it's due to a nutrient deficiency?
Some of these things, and the wheat leaf spot complex is probably one of the more challenging ones to identify. And I, over the years, have chatted with people like Miriam Fernandez at Current, who's one of our key leaf spot pathologists, and Jeannie Gilbert, who's retired in Winnipeg. And I would ask them, how do you tell the difference between septoria and tan spot or other things in the field? And the question was, it depends on whether they're producing very indicative symptoms. So with the septoria complex, it's the pycnidia. With tan spot, it's those cat eye oval lesions, often with a darker interior. chlorosis around the lesion. So if you look at nutrient deficiencies, and the main one that I would say in terms of wheat and barley is chloride. That's the one where chloride deficiency in wheat will produce symptoms that mimic septoria and tan spot. The key thing is that you may not get the extent of chlorosis around those that physiological leaf spotting due to chloride deficiency. I'm not sure about zinc. It's not one that I've commonly encountered as being an issue in terms of symptomology in cereals. It might have a subtle effect, but its chloride has been the more consistent one. And often the areas that look like tan spot or septoria have more an angular appearance to them. And that is one thing to keep in mind. If you're seeing those symptoms from the lower canopy and there's similar levels up to the top part of the canopy, that may also indicate a chloride deficiency. But because again, leaf spot diseases, not the rust, but leaf spots develop from the residue onto the early leaf tissue and then build. So you have a gradient of high levels of disease, lower, and then decreasing levels as you move up to those upper canopy leaves.
Yeah, good to know. Thank you.
Look at your soil test. Yeah, absolutely. In that situation, I'd be looking at pulling in as much information, eliminating potential for nutrient deficiencies and so on.
Soil, and I'm sure tissue test would paint a pretty good picture about what's going on as well. So the next question I have is, can you comment on bacterial leaf issues in oats? Like, what's the severity, what's susceptibility of oats? Can you comment on that?
Yeah, it's an excellent question, and we've had some interaction with POGA over the last few years, increasing concerns. So in oat, I think the primary one is halo blight. There may also be a leaf streak, so two different bacteria. It'd be very similar to the situation with barley and wheat and bacterial leaf streak, highly seed-borne in oat. Again, similar to the bacterial leaf streak in wheat and barley, there are no products to put on the seed. There are no products to put in crop. And as a consequence, seed infection will be an important parameter. If you're a long-term oat grower and maybe in a tighter rotation, there may be a role that rotation plays, but often the literature suggests that the bacterial pathogens in general don't typically survive as long as, let's say, the fungal leaf spots on the crop residue. So certainly they can be an issue. We've seen them in our oat breeding plots here at Lacombe over the last 30 years, and I would often get called out to look at what's there. Sometimes it was, is it manganese deficiency, gray streak, not bacterial in nature. If you've got a little mini microscope or whatever, one thing that you can do is take leaf tissue pieces, cut the sections out, put them in a clear container with water, and zoom in and look at streaming of bacteria from that cut leaf tissue. And if you see clouds of bacteria oozing out, you're dealing with a bacterial leaf disease. If that's not there, that's an indication that you're likely not dealing with a bacterial issue. So in oat, definitely look at what's happening in your crop. If you're seeing symptoms, you have them confirmed by the diagnostic lab in Regina, great resource that you have, or you send stuff up to Randy at the U of S. It's bacterial in origin. You might want to be cautious about the seed and reusing the seed off of that crop. There has been some work in the past with dry heat treatment in that 70 to 80 degrees C range for anywhere from 4 to 7 days. That's obviously not practical for commercial fields, but for the breeders and highly valuable small sources of seed, heat treatment might be a way of eradicating that pathogen and producing a source of seed that can then go into the foundation and pedigree and certified stream and not carry over that bacterial leaf streak pathogen.
Okay.
Or the bacterial issues in oat.
Yeah. Okay. Yeah, no, that's perfect. Thank you. The next question we have is, has there been any work done with strobe versus non-strobe fungicides on leaf disease and then for yield benefits specifically?
Yeah, there's been lots of work. We've worked with strobilurin fungicides over the years. Back in the early 2000s, when they were still research actives, and we still had the ability to do some spray and weigh testing with fungicides. We've moved away from that over the last 20 years. And yeah, definitely, they're a fungicide. They can be quite effective. They need to be put on before you have extensive disease development. Um, there was this concept of plant health benefit that was bandied about maybe 10 to 15 years ago. And in the absence of disease, and you hear messaging from the States and Europe, that plant health benefit may or may not carry over into actually producing a yield response. And we saw similar things with a seed treatment that included pyraclostrobin, where we saw under droughty conditions, the seed treated plots actually looked a bit healthier, a little greener. But when we looked at yield later on in the growing season, no difference. The one thing you want to be very, very cautious about with strobilurins is that if they're applied to head tissue, they will result not in a reduction in disease symptoms, but they actually, it's thought they introduce a stress on the fungus, and the stress response is to produce more DON. So with strobies are not recommended at heading. And unfortunately, there's research, including stuff that we've done in barley and work in the States and in Europe, where even at that flag leaf emergence stage, it seems to be having an effect where the grain that's harvested with strobilurins versus the untreated have a higher level of deoxynivalenol. So be very, very cautious about the strobies, especially as you're moving towards head emergence and post-head emergence.
Yeah, no, very good to know. Thank you. The next question we have is, does the bacterial ooze dry up at certain points during the day?
Absolutely. So I like to use the analogy of a glazed donut appearance. So if it's dry, let's say if it's been really wet, lots of oozing, and then it turns dry, those bacterial masses of cells will dry down. The water that's there will disappear, evaporate, and you'll get this sort of whitish or whitish yellow crusty appearance on the leaf tissue. If you angle that leaf, depending on the angle of the light, it can reflect. So yeah, that's another diagnostic feature. It's the oozing if you've got wet conditions, but if you're seeing that glazed donut appearance, that sort of crusty material, those are bacterial cells. That's the result of oozing. It's just dried down. So you've got a bacterial leaf disease issue there.
Okay, yeah, no, yeah, very good analogy there. The next question we have is there any leaf disease resistant varieties available in cereal crops?
Absolutely. So if you look at the variety guide that the Sask Ministry and the producer groups produce each year, whether that's Saskatchewan or Alberta or Manitoba, and I'm more on the barley side of things, and we've got some excellent information in terms of disease reaction. So you'll have columns for spot blotch resistance, net form, spot form resistance, scald resistance, mostly in the Alberta guide, I might add. So there's some excellent information there to help with the producer's variety choice in maybe targeting. They've had issues with, let's say, net form, net blotch. So the barley side of things, a much clearer picture. Obviously, there's resistance for other issues like loose smut and covered smut and FHB. And wheat, it's a bit of a different picture. So you've got good information on FHB, the rust complex, bunt, but very limited to poor information on leaf diseases because it's not a priority 1 disease for the Prairie Recommending Committee on Wheat, Rye, and Triticale. So we don't produce leaf disease ratings. And when we did, often it was, if it was there, and it was referred to as the leaf spot complex in wheat. Didn't differentiate whether it was tan spot or the septoria complex. So that's a definite hole that we need to look at, or a limitation that we need to look at addressing. And I've emphasized that to my wheat colleagues over the last 25 years, that we need to ramp up things in terms of wheat. So yeah, there I would talk with the seed retailer, perhaps the seed grower might have some experience in what their experience has been over the last few years and comparing varieties on their farm, and they might see that some varieties show more tan spot or septoria versus others. So unfortunately, that's the information you have access to. The variety guide doesn't have that.
Okay, perfect.
Unlike barley.
Yeah.
That's...
The next question we have here is, if there's no in-crop treatment for bacterial leaf streak, what management strategies would you suggest for reducing the risk of bacterial leaf streak?
Well, it's as much of an integrated approach as you can use. So I would look at your past history first of all, and if you've had issues with it, and then potentially if you're using seed from that field. But regardless, I would be having that seed, whether you're using it certified or not, have it screened for bacterial leaf streak. And depending on the presence and level, this is the challenge that Randy Kuchar is trying to address, and we've been running trials for him here at Lacombe and a seed threshold trial. We can get different levels of disease, but when we look at yield differences, we don't necessarily see a response. So different levels of seed infection resulting in different levels of disease, and so on. But seed is an important parameter. So avoiding, let's say, seed lots with high levels of seed infection. And then I would look at varieties. And unfortunately, we've tried to get access to some of the varieties that are in the regional trials, but there is a reluctance amongst the seed trade and so on. I tried to do that here in Alberta. I approached RVAC. It's equivalent to your variety, regional variety system in Saskatchewan, and yeah, it was a no in terms of providing seed. So what we have is the work that Randy has been doing, and we're serving to help with that. Some of our breeding work that we're doing with the breeding programs. So you'll see there's some differences there. So things like CDC Copper, Austenson, and others, and there's genetics coming through the system for all three programs that are looking pretty exciting as far as variety resistance. The other thing that I would look at is where you're located. And if you're an irrigated producer, and here in Alberta, southern Alberta under irrigation, and I would say in areas where you get more frequent severe weather systems, this disease is going to be promoted and spread via severe weather events, strong driving rain, perhaps some hail, strong winds that cause a lot of leaf rubbing and abrasion and damage that provide openings for the bacterium to get into, and frequent rainfall or frequent irrigation will drive this pathogen. So be cognizant of that. Maybe if you're under irrigation, if you can cut back on the frequency and amount, obviously not compromising the ability to meet the water needs of that crop, that may help.
Okay, perfect. The next one is, over the past number of years, I've seen that with tight rotations, the disease pressure is higher and has a bigger impact than the environment itself. Are you seeing or hearing the same observation?
I, as a pathologist, and working in this as whether as a summer student, a grad student, or a research scientist, the weather is the key factor. However, if you look at that disease triangle and one or more of the components, so in this case, the environment may be not as favorable, but if you've got a highly susceptible variety and on a tight rotation, continuous wheat or continuous barley or wheat, canola, wheat, you may have enough inoculum buildup that even though the weather is, it's not so dry that nothing's going to develop, but that tight rotation, lots of infected residue, and a highly susceptible variety can compensate for that slightly less favorable weather conditions. So you may see development of disease. At most, it might be moderate in nature. But again, these things can respond to later season showers during that critical grain filling period. So you can have some infection occurring there. But in general, if you have tight rotation, susceptible variety, slightly not as favorable weather, but again, keeping in mind there's a critical point where it's so dry nothing's going to develop. And the one aspect I want to talk about, which I didn't during the presentation, I did mention it to Ali, is we've talked about above ground disease issues. We need to look at below ground, and often in those scenarios, The worst case scenario is moisture earlier in the season to facilitate early infection of the root tissue. So things like common root rot, the dryland or crown rot fungus, take all. And then what happens is the most significant yield losses occur when you get dry conditions during the grain filling period, because that compromised root tissue, there's not enough moisture and it can't keep up with the water and nutrient demands of that crop. So in that situation, a drier finish to the year, starting in mid-June throughout the rest of June and into July, may cause a higher level of yield loss if you've had moist conditions earlier on that establish infections early. Seed treatments, people probably wondering about that. Just one other point, do not provide good management of root diseases. We've looked at seed treatments for common root rot and barley. The reason being is that if you look at movement of that seed treatment, most of it's staying on the seed itself, or it's moving into the soil matrix immediately adjacent to that seed. The least amount of fungicide goes into the root tissue, very little. There is some fungicide, more fungicide that actually go up into the first true leaf, second true leaf, and so on. So don't assume seed treatments are going to address that root disease problem for you. Absolutely, seed and seedling blights due to Fusarium graminearum and things like the spot blotch fungus, whether seed-borne or in the case of the spot blotch fungus, soil-borne sources of inoculum, very important in terms of managing seed and seedling disease issues.
Yeah, you beat me to the seed treatment aspect of that. The next question we have here is related to seed treatment. So this question asks, how can seed treatments aid in later development of disease?
Well, that's a good question. So I visited Australia and Mark McLean back in 2012, and we were there early October, and we were looking at some of his plots, and I said, geez, that fungicide applied in crop? And he said, no, it's seed treatment. And it was a product, fluxoxypyrad. So it's in some of BASF's products, foliar and seed treatments, I think FX4. It was a standalone product used in Australia at a much higher rate than is used in the mixtures that we see in seed treatments here in Canada. It's extremely mobile, so it readily moved into the first true leaf, second true leaf, and third true leaf. So it had an effect. We did some work in the early 2000s with seed treatment and saw some responses in some years where there was wet conditions earlier on, facilitating disease development. But in years where things were dry earlier on, which may have affected fungicide movement into the leaf tissue perhaps, but where wet conditions occurred closer to flag leaf emergence and later, there was no effect of the seed treatment. So you need to look at the response. The other issue in Australia, fast forward to 2017 when I visited, and then after that, they were seeing widespread resistance in the net-form net blotch fungus to Xenium or Cystiva, which is single active fluxoxypyrad at a higher rate. So I would be using a combination of seed treatments. We've done some work here, and there is some potential benefit that can potentially persist into seeing a later season impact on leaf disease. So it reduces the amount earlier on, that slows development down, and then we can see a yield response. But it's under particularly specific conditions where I think you've got tighter rotation, susceptible variety, and where you get early season moisture that drives that fungus to develop following first leaf emergence through to maybe the 3 or 4 leaf stage.
Okay, perfect. We're getting down to the last few questions here,
Sure.
but about
Yeah,
3
no
more.
problem.
The next question is, should I continue spraying tilt with in-crop, or is it detrimental? Is a full rate of tilt worth it, or should I cut the rate in half essentially, or lower the rates?
So looking over my shoulder, I'm not advocating reduced rates. And that's something that, the concern with reduced rates, with fungicide resistance development, the things that really drive it are frequent use of the same actives and as a single active. So the more times you spray within crop, the more times you spray between years will facilitate that that development. Sorry, can you repeat the question? I'm getting old and forgetful here.
Yeah, no worries. The question was, should I continue spraying tilt in crop, or is it
Yeah.
detrimental to the crop? And then they asked about the full rate versus the half rate.
Yeah. Well, tilt, a lot of the fungicides go through a very rigorous process in terms of development, evaluation, and final registration. So they will look at things like phytotoxicity. So, I don't necessarily see issues with the products we have, maybe potentially with some surfactants. The most frequent issue we see with phytotoxicity is where producers will mix in something like a foliar copper product in with their tilt. And I'll get calls where the farmer says, my leaf disease is higher where I've sprayed. And the first question I ask is, did you add anything in with the fungicide? And invariably it was yes. Now this is 15, 20 years ago. Our formulations back then were not like we've got right now. So I would be more concerned about phytotoxicity associated with foliar fertilizer products. So be cautious about that. Work with the retailer or supplier to look at the conditions and so on. Half rates in general, the concern is that it can contribute to potential risks with fungicide resistance development. It's more the frequency of use within and between growing seasons, but half rates will allow a larger segment of that pathogen population to survive. And with a lot of the products like triazoles and SDHIs, it's not an on-off type of resistance. It's a slow progression from being fully effective to starting to see a reduction in effectiveness to, if you look at Australia or Europe, where those products are no longer effective. The targets may be a little more complex, so it's a little harder for the fungus to overcome that active, whereas in contrast, the strobilurin is a very specific target site, and this is where we've seen issues. If you go back to the early 2000s with headline or pyrrolactostrobin, some of the other strobilurins that were introduced into the marketplace, and producers started using them frequently 2 to 3 times in the growing season for ascocutta and chickpeas and lentils. And lo and behold, they had, and even microspherula and field peas, they've had resistance development. So yeah, that half rate definitely is tempting for people who've looked at half rates over the years, perhaps under certain circumstances, a reduction in risk, but where you're still concerned, you want to cut your input costs, but it's not on the label. And what you could do is look at a product that has a range of rates on the label and use the lowest labeled rate to help cut the cost in terms of fungicide application. But tilt, still an effective product, not great for FHBs, never been recommended for FHB. But it's rust, the leaf spot complex. But again, look at what your use patterns have been over the last number of years. If you've used that half rate in with a herbicide, invariably you'll have to go back in and spray again at flag leaf emergence if there's a leaf disease risk there to protect those key upper canopy leaves. That's where you start seeing a shift. And we have seen back in the early 2000s, we did some work with net form and spot form and across the prairies, and we had isolates of the net form, net blotch, and spot form blotch fungus that were resistant to propiconazole, the active ingredient in tilt, and some of the generic alternatives. So it's one that has a long use history dating back to the late 80s, I would guess. And it's a product that's been used as a half rate in with the herbicide, thinking that it has a benefit. And I would argue, based on the chemistry of the fungicides we used, the movement in the plant, the tissues that you actually need to protect in terms of green filling and yield, that early application is not something that I would typically recommend.
Yeah, no, very good advice. Cutting rates of any pesticide product can really be a slippery slope. So the next question we have is, have you studied the effects of heavy harrowing in the fall to help break down the leaf disease triangle?
Well, that's an excellent question. And, when I was a young scientist, when I had a little bit more hair and I was skinnier, Not a lot more hair, by the way. Back in the mid 90s, there was a huge amount of work being done by Ag Canada scientists across the prairies, looking at that transition from direct seeding or from conventional tillage, so tandem disking, heavy cultivation and so on, harrowing, and comparing that to direct seeding. And what people at Karen Bailey and Guy Lafond at Indian Head, I think Adrian Johnson and Randy Kuchar were finding at Melfort, what we were finding here in Alberta and central Alberta and the Peace, is that tillage wasn't necessarily as important a strategy in terms of disease management. And if you look at the first outbreaks of FHB in the mid 80s in the Red River Valley, the tillage practice in those fields was 2 tandem discs in the fall, or maybe one in the fall and one in the spring, followed by cultivation and harrowing.
Okay.
Very heavy conventional tillage did not prevent that development of that disease. And by 93, that was the watershed year. In those fields, corn was either the previous crop or was a previous crop and part of the field. It's the exact same scenario with virulent blackleg in northeastern Saskatchewan. My dad, I spent time on a tandem disc in the fall and cultivation. We had lots more summer fallow. It absolutely did not control blackleg. So my own view based on the research is that the weather is the key factor, the level of variety resistance, and the rotation you're using. Those 3 factors, along with seed infection, are the primary drivers for risk. The issue sometimes, and if you look at biology of these things, and we're losing that foundational knowledge and expertise, you look at the nature of these fungi, a lot of them, if the substrate they survive on is high in nutrient content, they often don't produce the sexual fruiting structures. Either they don't produce them or the level is lower. But if that substrate, the nutrient content decreases as a result of decomposition and whatnot, that triggers a response in that pathogen. My food base is disappearing. I need to find a living host. So it triggers the production of spores, fruiting structures and spores. So sometimes, there is a worry, at least in the past, we were concerned about minimum tillage or maybe even light harrowing that might cause a little bit more decomposition, not as much as, let's say, if you're using moldboard plowing, and that might actually trigger more fungal spore development. That's, you look at lab studies with certain fungi where they're growing on a rich nutrient media, no production of perithecia or pseudothecia. Once that fungus hyphae grows into another part of that petri dish with a low nutrient substrate, bam, those perithecia and pseudothecia are produced. So the harrowing, that might be useful in terms of residue management and getting good seed to soil contact and less interference from that residue in terms of the seeding operation. But I would look at other factors like what crops have I grown previously, what's the weather conditions going to be like or following them during the course of the growing season, and is my variety susceptible or not?
Okay, yeah, no, very very good answer. We appreciate it, and that was the last question. So
Okay.
I'm gonna put the QR code back up on the screen for any participants that might have missed it earlier, and just thank you again for joining our webinar today. You gave a really good presentation as you always do, and we just want to really say a special thank you for all you've done for the
Awesome.
industry over your career, your approach to research and sharing it. It's so practical for producers and agronomists, and we really appreciate learning from you and just all the work that you've done and the impact that you've had on the industry.